Have you started recording?
Yes.
Hold on 1 minute. Okay. Welcome to today's episode of Hephzibah's Talks. My name is Hephzibah, and today we're going to be dealing with tools and techniques in the field of biotechnology. Like every trade, biotechnology has its own tools and techniques. Now, they are so numerous in number, I cannot break them all down to you today, but I will be taking them one by one from time to time. Today, we're starting with recombinant DNA technology, or rDNA technology. Recombinant DNA is the manipulation of DNA material or DNA molecule in new combinations to make up brand new genetic materials. I will be simplifying that for you in 6 simple steps. Step 1, isolation of DNA. Okay, so DNA is taken from its host and separated into a culture medium. So isolate means to separate it from others. So it is separated usually in a culture medium. Step 2, we have cutting DNA. Now, don't you think, or think not thou, that when I mean cutting, I meant with scissors. No, we don't do that here. There is a molecular material used to divide at specific fragments in specific locations of the DNA, and that material is known as restriction endonuclease enzyme. Simply put, restriction enzymes. They cut the DNA material at specific locations in specific fragments. Step number three, we have insertion of foreign DNA. Now, DNA is taken again from a different material, a different species altogether, and they try to incorporate it into the host DNA. They also cut this foreign DNA using the restriction enzymes so that it can produce something we call sticky ends. Now, the host DNA and the foreign DNA are joined together at specific locations with the fragments. This is not yet a living molecule, mind you. And that brings us to step number 4, joining the DNA. Now, though they are stuck together, they aren't technically joined together. And this is why we call this joining the DNA together. Now, there is a molecular gum we use for this, and it is called ligase enzyme. The ligase enzyme fills in all the cracks in the DNA and seals it up so that now it can become a living molecule. Step number 5, replication of DNA. Now, every living thing has the ability to express and replicate itself. So this DNA, recombinant DNA, has the ability to express itself through 2 major parts for living organisms. For bacteria, it is called transformation, and for eukaryotic cells, it is called transfection of DNA. As the host cell replicates, the foreign DNA begin to also express themselves, resulting in the production of protein encoded by the foreign DNA. And the last step number 6, is selection and analysis of DNA. By now, we have reached the final step of the recombinant DNA technology. And the techniques used for this is called antibiotic resistant genes and reporter genes. Now, these cells that have finished the process of recombinant DNA are selected and analyzed for their desired traits and protein production. I hope you've learned a thing or two from DNA technologies today. I want you to also know that DNA has shaped so many fields, such as the medical research, biotechnology, and agriculture, in the production of pharmaceuticals, biopharmaceuticals, in protein production, and the production of genetically modified organisms. Also note that recombinant DNA can otherwise be called genetic engineering. And to me, that sounds like mutation. But trust me, these are one of the tools used in gene editing and expansion. Thank you for watching today's episode. Don't forget to like, share, and subscribe to my YouTube channel. Bye-bye.